Equine Juvenile Spinocerebellar Ataxia, often abbreviated as EJSCA, is a fatal neurological disease affecting quarter horse foals. And until recently, it was one of the most heartbreaking and mysterious conditions veterinarians had ever encountered, because healthy young foals that appeared completely normal at birth could suddenly begin losing coordination, struggling to stand, and rapidly deteriorating with little warning and no available treatment. Now, researchers at the University of California, Davis have identified the genetic mutation responsible for equine juvenile spinocerebellar ataxia, a breakthrough that not only solves a medical mystery that has puzzled veterinarians for years, but also gives horse breeders an opportunity to prevent future foals from suffering from this devastating disease before they are even born. For breeders who have watched a promising foal deteriorate despite receiving every possible veterinary intervention, this discovery represents something incredibly valuable: answers. And for the future of the American quarter horse breed, it could represent the difference between continuing to lose foals to an invisible genetic threat and finally having the tools necessary to stop it. Because the reality is that some of the most dangerous diseases affecting horses are not the ones that arrive through infection, injury, or environmental exposure. Some of the most devastating diseases are hidden quietly within the genetic code itself, passing unnoticed from one generation to the next, until the right combination of genes suddenly produces a tragedy that nobody saw coming. That is exactly what happened with equine juvenile spinocerebellar ataxia. For years, isolated cases appeared across the country, leaving horse owners, breeders, and veterinarians searching for answers as young quarter horse foals developed severe neurological symptoms that seemed to emerge out of nowhere, progressing rapidly from subtle coordination issues to profound weakness and ultimately the inability to stand, forcing owners to make heartbreaking decisions about foals that only weeks earlier had appeared healthy and full of potential. What made the disease especially frustrating was the fact that affected foals often looked perfectly normal at birth, showed no obvious warning signs during pregnancy, and came from breeding programs that had produced healthy horses for years, making it extraordinarily difficult for breeders to understand why these tragedies were occurring or how they could prevent them from happening again. The mystery began attracting attention to veterinarian researchers in 2020, when a quarter horse filly from Oregon developed a pattern of neurologic symptoms that did not fit any known disease. At first, veterinarians suspected more familiar causes of neurologic dysfunction, including infectious diseases, developmental abnormalities, trauma, or previously identified inherited disorders. But as testing continued and more information became available, it became increasingly clear that they were dealing with something entirely different. What initially appeared to be a single, unusual case gradually becoming something much larger. As additional foals were identified and researchers began comparing pedigrees, medical records, and genetic information, a troubling pattern started to emerge. The affected foals appeared to share ancestry, suggesting that the disease might not be random at all, but instead might be inherited, hidden within bloodlines that otherwise produce successful, athletic, and healthy horses. That possibility launched an intensive investigation led by Dr. Carrie Fino and her team at UC Davis, one of the world's leading institutions for equine genetic research. And what they eventually uncovered would become one of the most important discoveries in modern equine neurology. Using advanced genomic sequencing technology, researchers identified a mutation in a gene known as FDXR, a gene that plays a critical role in cellular function and nervous system health. The discovery was remarkable, not only because it finally explained why these foals were becoming sick, but also because the mutation was located in a non-coding region of DNA, an area of the genome that scientists once believed was relatively unimportant, but which is now increasingly recognized as having profound effects on how genes function. The consequences of this tiny genetic change are devastating. Foals that inherit 2 copies of the mutation develop progressive degeneration of axons, the nerve fibers responsible for transmitting information throughout the nervous system. As these pathways begin to fail, communication between the brain and the body breaks down, causing affected foals to lose coordination, balance, and ultimately their ability to stand and move normally. For horse owners watching the disease unfold, the experience can be almost unbearable because the progression is often incredibly rapid. A foal that appears healthy one week may begin showing subtle neurologic abnormalities the next, and with a surprisingly short period of time can deteriorate to the point where humane euthanasia becomes the only compassionate option. Yet, despite the tragedy associated with equine juvenile spinocerebellar ataxia, there is now a reason for optimism. For the first time since the disease was recognized, breeders have a way to identify carriers before breeding decisions are made, allowing them to dramatically reduce the risk of producing affected foals while still preserving valuable bloodlines and maintaining genetic diversity within the quarter horse population. And that is why this discovery matters so much, because this story is not simply about a rare genetic disease. It is about how modern veterinary science is transforming horse breeding, how genetic testing is becoming one of the most powerful tools available to breeders, and how a handful of mysterious cases ultimately led to a breakthrough that could save countless foals for generations to come. Thank you so much for joining us. And remember, I am not AI. If you listen closely, you can hear me breathing.